once inside the cell, it's not truly inside the cells. I'll go into in a little more detail. It's picked up in a vesicular trafficking pathway, and how do you get that out of the vesicular compartments to the cytosol and then to the across the nuclear membrane barrier. These are, from a drug delivery standpoint, daunting challenges, especially when you begin to add them up. There have been two major approaches then to dealing with these barriers. The first is to use cationic polymers to condense the negatively charged DNA. And because these are polymers, the resulting condensed particle is called a polyplex. The other major approach is to use a cationic lipid, and sometimes this term liposome is used. I have it on my slide because it's used so much, but it's not actually a very accurate description of the structures of the resulting complex. It's not very well described exactly what the structure is, but probably not liposome. Nevertheless, the cationic lipids have been the other major category. They share in common the positive charge that interacts electrostatically with the negative charge on the backbone of DNA. And the condensed particle in this case is called a lipoplex. And so you'll see lots of different companies out there that are trying to develop polymers and polyplex technology, and another category of companies that are working on cationic lipids and lipoplex technology. The key point is that both of these agents, when they condense the DNA, they sequester the DNA and protect the DNA against nucleases. To some extent, if they're designed appropriately, they can also protect against serum inactivation by the binding of different serum components, although that remains a serious problem and detail that you have to worry about in the design of either your cationic polymer or the cationic lipid. And then, in addition, they condense the plasmid DNA into very small particles, typically under 200 nanometers in size. Some people say that you need to be under 100 nanometers in size, but the general point is that the condensation allows the DNA to be taken up with much higher efficiency. Looking at the cationic lipids first, there is a whole literature on different cationic lipids, and there's a whole variety of biotech companies that have their favorite and protected cationic lipid compositions. The main game here is to get efficient condensation without toxicity. And this has been a major issue with the cationic lipids, is toxicity. In vitro, this is maybe a little less of an issue because you can take some hit in toxicity and still get your targeted cell population. But in vivo, this has been a major problem and a barrier to the clinical development of cationic lipids. One interesting point in regard to this that is, again, part of the challenge of doing this with high efficiency is that on the one hand, the first thing that you want to do is condense your DNA very tightly so that it's sequestered from nucleases and from serum components. But you have to realize that once it gets inside the cell, it has to decondense so that the DNA and plasmid can get to its target and be in a form that's accessible for transcription. And so these two design criteria go exactly opposite to each other. On the one hand, you're wanting to condense very tightly, but in the other case, you're wanting to decondense. And there's major effort to design into these systems degradability so that it condenses outside the cell, but through some mechanism, reduction or cleavage, once inside the cell, you can decondense the DNA from the degraded carrier. This is a cartoon of in this case, sort of a liposomal cartoon of how these one of these cationic lipids can be used to condense DNA. And so you're seeing particles again on the size scale of about 100 nanometers. This should be viewed purely as a cartoon. The actual structure of these complexes it has not been so well described, and it's very dependent on the type of lipid and the ratios of the lipid to DNA. And in my opinion, this is a detail that needs to be considered because it plays a big role in the decondensation process that ultimately has to occur inside the cell. This is a cartoon for the polymer, cationic polymer approach. And I guess Ringsdorf really was the first to propose this kind of picture, this advantage of polymers as carriers. The nice theoretical advantage of polymers over cationic lipids as a carrier is that you can build in additional functionality. And so you can have the cationic charges along the backbone that condense the DNA, but by relatively simple grafting approaches or incorporation of specially designed monomers, you can add additional functionality. And some of the most obvious things that people have been trying are to add in peptides that represent nuclear localization sequences that can address that barrier of entry from the cytosol to the nucleus. There can be additional agents that act to get the carrier, once it's taken up into the vesicular trafficking pathway, get it out of that pathway, usually from the endosome, and thus get over that barrier of entry to the cytosol. You can also add in targeting agents that stimulate uptake more efficiently than penocytosis so that you can get receptor-mediated endocytosis. And you can also think about adding in PEG groups, are the most common groups, that sterically stabilize the complexes against serum inactivation. And this has been a huge problem, this serum inactivation for the cationic lipids, but it's been shown now, I think, and there is a lot of literature and clinical experience now with pegylated polymers that show that this can be a very important component in aiding the escape through the reticular endothelial system for systemic delivery, or locally to control nonspecific uptake of the particles if you have a targeting agent that can target the appropriate cell that you're after. A couple of examples, the sort of first generation polyol-lysine and polyethyleneamine are examples of cationic polymers. And in fact, most of the work has been done with simple cationic polymers that don't have the more sophisticated elements. Polyol-lysine is not really a viable candidate anymore, I think. For in vivo work, there's a lot of toxicity associated with that composition. PEI is still used a lot, and there are a lot of names for these different companies, like Gene Jammer that Steve was talking about, the different companies that produce silencing RNA have their own little names, but a lot of these polyamine polymers are in fact modified versions of PEI. And PEI, the details are very important. It gives you one idea of how you can get these structure-function relationships going on the delivery side. There are highly branched versions of PEI versus linear versions. That branching versus linear has been shown to be an important detail in their efficiency, but also their toxicity, and it's usually a trade-off between those two. And also molecular weight has been shown to be very important, both from the standpoint of toxicity and efficiency. But this is really certainly a very active area, both in academia and in industry, is trying to come up with better polymer compositions. With antisense, moving to the other two types of nucleic acid drugs, basically antisense is a method for inactivating a specific mRNA species at the mRNA level. And so it basically just interrupts the central dogma at the mRNA stage. And these are short pieces of DNA that hybridize through complementary base pairing to target mRNA sequences. They're usually chemically modified, so they're not as susceptible to nucleases. This is one of the nice things about antisense drugs as compared to plasmid-based drugs, is that you can conveniently chemically modify them. That chemistry has been pretty worked out at this point in development. There are a few competing modification chemistries, but they're out